Hello, my name is Sam Westrake, and this is the third tutorial video for the Standards Challenge on Mosaic. In this video, I will show you how to explore the results of the sequencing data and the protocol metadata that you've provided for the Standards Challenge. We'll begin by navigating to the challenge. In earlier videos, I showed you how to request samples using this tab on the left-hand side titled Request Samples. I also showed you how to submit your data in this Data Submission tab, including how to upload your sequencing files to Mosaic, and how to provide your metadata and protocol details using the Submission Manager. Now, we're going to go to this fourth tab called Results Exploration to see our results. Here, the results are visible for all contestants and are split into two groups. In the Protocol Statistics tab, we're able to see the differences in the protocols. These are differences in the metadata that you provided. This does not look at the organism results. In the Analysis Results section, we can compare the organism annotations that were derived from the sequencing files you uploaded and see how those compare to the steps that you made in your protocol. We'll begin with Protocol Statistics. Here, we can see a breakdown of how many participants selected each option in the protocol's metadata for each of the questions. We have some suggested charts, including those that are amplicon specific, covering only the 16S results, and some general charts that cover both the 16S and the whole genome results. When we click one of these links, it will pre-populate the information here, and when we scroll down, we can see the number of responses for each category for that question. Here, for read length, we see that some of the participants provided 250 base paired end reads, and some of the participants have provided 150 base paired end reads. For each of these, it looks like about 42 samples were provided. We can change the data source to see either the results for all answers, the results only for those who performed 16S sequencing, or to see the results only for those who performed whole genome sequencing. In this case, they're about the same. We can also change the metadata category and the question that we select. From sample storage, perhaps we want to see how samples were typically transported in the lab. When we select that, and we summarize this by both the DNA and the stool results, we're able to see that pretty much everybody has transported their samples at room temperature. We can narrow this down and select only the stool results to see how many people transported their stool results at room temperature or using another method. These results will change as more participants come and provide their data, so be sure to check back regularly to see more answers appear on this chart. Now, let's go back to the main results page and we'll take a look at the analysis results. Now, let's take a look at the analysis results to see how our organism annotations from our samples compare to those from others. When we click here, it will take a moment to load because of the visualization, so have some patience. Here, on the analysis page, we can see a PCOA plot generated with Emperor that shows the difference between the different submissions. We can also see an alpha diversity plot which shows us the calculated overall alpha diversity for each of the samples within the kit. As you can see, for each of the 7 samples, there is a wide degree of variation that's been observed from all the responses so far. Let's focus on the PCOA plot. Here, when we scroll down, we can see how the different samples compare to each other. You will note that the sample names are anonymized. If you answered all of the preferred questions when you were filling out your metadata, you will find a box at the top of the page which tells you which of these anonymized submissions is your own. Remember, this information can only be provided before you click Submit. On this Emperor chart, we're able to click and drag and change the direction of our PCOA plot. Under Colors, we're able to select which question we use to color our points. If we select, did you perform amplicon or whole genome sequencing, for example, we're able to see that 14 of the points shown here use 16S sequencing, almost all of which are clustered in a very small dot here, and 7 of these points used whole genome sequencing. those points are more scattered. You can select any of the metadata questions, including ones such as how is the indexing and barcoding performed, how were samples stored, the DNA template concentration measured, and any others. You can also select sample type to split by DNA versus feces. And we see that all of the DNA results cluster pretty closely over here, while the fecal results are more scattered. This plot is currently showing the responses for all approaches, both the 16S and whole genome sequencing approach. Under data source, we can narrow this plot down to view only the 16S results or only the whole genome sequencing results. If we select 16S, the plot regenerates and we have fewer data points to display. Once again, we can change the graph around, and under colors, we're able to color these points by any question. In this case, because we have selected to draw our data only from the 16S results, all of them are colored red to show that they performed amplicon sequencing. If you wish to capture this plot and save it, under Options, you can find the possibility to create a screenshot. You can save this as either an SVG or a PNG image and use this however you choose. This concludes our explanation of exploring the results of the Standards Challenge. If you have additional questions, please feel free to contact us at info@mosaicbiome.com. We will be regularly releasing data dumps of all of the information that's been provided so far, with all pertinent information anonymized, so you will not be able to track individual submitters by the results files that we provide. Until then, thank you for participating in the Standards Challenge.